Good afternoon, everyone, and welcome to Genome Webinars. I'm Ben Butkus, Editorial Director at GenomeWeb, and I'll be your moderator today. The title of today's webinar is The Promise of Liquid Biopsy for Individualizing Cancer Care and is sponsored by Agena Bioscience. Our speaker today will be Klaus Pantel, Chairman of the Institute of Tumor Biology at University Medical Center Hamburg-Eppendorf. He will be followed by Alexander Sartori, Senior Manager of Scientific Affairs at Agena Bioscience, who will provide a brief overview of Agena's mass array system and variant detection panels for circulating cell-free DNA. Attendees may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Klaus Pantel of the Institute of Tumor Biology at University Medical Center Hamburg-Eppendorf. Please go ahead, Dr. Pantel.
Well, it's really my pleasure to speak here today about circulating tumor cells and liquid biopsy. And I think it's really fantastic to be here today and to give you an overview of how circulating cell-free DNA in blood is current biomarker for liquid biopsies. This is my disclosure information. Liquid biopsy, the definition is that liquid biopsy is the analysis of cells or cellular products, and in particular the cell-free DNA has become a prime liquid biopsy marker. And most of the studies focus on blood, but of course also other body fluid can be assessed for circulating DNA or circulating tumor cells. The vision here is to get a more comprehensive and real-time information on the tumor burden by the analysis of blood samples, and particularly also sequential blood samples. Well, liquid biopsy is complementary to tissue biopsy. Tissue biopsies are invasive and some locations are difficult to access, for example, in the lung or the brain. Single biopsy can miss relevant tumor clones due to intra-patient tumor heterogeneity. And of course, sequential tissue biopsies in individual patients can be used for real-time monitoring of therapy responsive, and that is, of course, less feasible with sequential tissue biopsies. So there are several advantages how liquid biopsy is really complementary to tissue biopsies. Well, in terms of what are the sources of cell-free DNA in the blood? Well, in the beginning and early cancer stages, the primary tumor is the key source of circulating cell-free DNA. At later stages, regional lymph node metastasis can contribute to the circulating cell-free DNA. And in even more advanced stages, when there are solid metastases, the distant metastasis, for example, in the brain, in the liver, or in the bones, can contribute to the circulating cell-free DNA and the tumor cells. And we have really reviewed this, I think, topic, and some of the reviews are shown here on that slide for further information. Well, as I said before, the blood is really the key fluid that we investigate for circulating tumor cells and tumor cell DNA. And in the cellular fraction, we can look at cells, but today I will focus on the plasma fraction, where we find the circulating tumor DNA. But of course, we also find other circulating markers such as RNAs, proteins, extracellular vesicles, or tumor-educated platelets. But definitely the cell-free DNA has become the prime target of liquid biopsy analysis. Well, in circulating tumor DNA, of course, is diluted in a sea of normal DNA. And there are various ways how we can identify that the DNA is derived from tumor tissue. We can look at point mutations, we can look at amplifications, we can look at fragmentomics, or we can look at epigenetics marks, just to mention a few ways how we can identify the circulating tumor DNA in the sea of circulating normal DNA. The tumor DNA is usually in the blood plasma at a low concentration. In patients with early disease, this concentration might be less than 1%. In patients with advanced metastatic disease, this concentration might be above 1%. So depending on the stage of disease, we need more or less sensitive methods to detect the tumor DNA that is diluted in the sea of normal DNA. And that's very important. And I think we have to be careful if we use hypersensitive methods. We may reach a background of mutations that is, for example, derived from leukocytes, and these SHP mutations, of course, need to be subtracted from the real tumor somatic mutations that we want to analyze. Together with Ergina, we have developed, for example, a panel that is NGS-free, and I think Alex Sartorio will talk about the technology in the next talk. And here, for example, we have developed a panel for the investigation of melanoma patient. We have investigated 13 genes that are relevant to melanoma treatment and progression, for example, BRAF mutations. And overall, the panel includes 65 mutations. And the technology is rather sensitive. It can go down to 0.1% mutation frequency detection, and the results will actually be there within a few days without any complicated bioinformatics. We also went on, besides looking at mutations with this technology, to look at methylation-specific detection. Epigenetic marks, of course, are super interesting in terms of identifying the tissue origin of the cell-free DNA in the blood, but also there are other applications where epigenetic marks become very interesting, also in terms of early cancer detection. So here we have developed a panel of different methylation-specific marks and at different genomic locations, and just to show that this can be also applied for that purpose. This is just a non-published set of data that we have actually achieved together in collaboration with Ergina. We have looked at healthy donors and carcinoma patients, and as you can see here in this very small population of patients and control, we have a sensitivity of 92% and a specificity of 95%. But of course, we have to analyze larger cohorts to validate these very promising findings. And in this case, the cancer patients were triple-negative breast cancer patients. In order to really get this into clinical practice, a liquid biopsy, we have actually established a few years ago a EU-funded network called Cancer ID, and that was a public-private partnership between academic institutions and companies, including Ergina. And we had, at the end, 40 partners from the European Union. We were focusing on lung and breast cancer, focusing on circulating DNA and CTCs, but also included circulating microRNAs. At the end of this funding period, we wanted to sustain this very fruitful network, and we actually established a new consortium called the European Liquid Biopsy Society. And we actually followed up the philosophy of Cancer ID. I think we started with technical evaluation of method, which is very important to get a good technical standardization. We established guidelines and SOPs, and then we continued with the clinical validation. And the goal here at the end is to get really methodologies that are ready for clinical use and can also get support of regulatory approval in the future. Together with the Cancer ID group, we actually validated different liquid biopsy assays, including circulating tumor DNA assays, and we published these validations and guidelines in clinical chemistry in several papers in recent years. We also performed within Cancer ID a ring trial where we looked at different ctDNA methodologies, and as you can see here on that slide, we also had included the mass spec technology from Ergina with very nice results compared to other technologies. Of course, in order to go on with this activity, we have established the European Liquid Biopsy Society, And here the goal is definitely to foster the introduction of liquid biopsy into clinical practice. There is still a gap between wonderful publications, but not many new biomarkers in the clinic. So we want to close this gap. Here we want to encourage the interaction between academia and industry, which is important to get kits available for these analyses, provide a better partnership with regulatory agency, healthcare providers, and patient advocacy group, support the implementation of liquid biopsy into clinical trials, develop guidelines and provide training for liquid biopsy, disseminate the knowledge that these methods exist for liquid biopsy, and increase the visibility of Europe as hub for liquid biopsy. And we have also reached out to the US by being a founding member of the International Society for the National Alliance for Liquid Biopsy Analysis. The structure of the ALPS is shown here. We have 4 working groups on dissemination and education, then on infrastructure and standardization, a 3rd working group on the clinical studies, and a 4th working group on the technologies. And if you're interested in this, please drop me a mail and you may become a member of this consortium. What are the clinical applications of ctDNA analysis? Well, the first application is early diagnosis and early detection of cancer and screening for cancer. The second application is, of course, to get information on the response and follow up the patients after the initial treatment to see whether the patient has been cured or not. The third application is to analyze tumor evolution, in particularly resistance mutation to the therapies that are applied, and learn how they can be overcome by additional therapy. And the fourth application is detection of minimal residual disease. That means that there is residual tumor burden that is not actually detected by conventional imaging procedures. And we use now the circulating DNA to see whether this residual disease can lead to relapse in cancer patients. Here it is shown in a graphical representation. And one of the stages of tumor development that is very exciting for the analysis of liquid biopsy is really the stage of minimal residual disease, as shown here in the middle of the graph. You can see that in some tumor patients, we are not able to cure them and the disease may come back, but it may come back to a level which is not detectable by current imaging procedures, but can be detectable now by circulating DNA analysis. And this detection of MRD, or post-surgical molecular relapse, allows us now to design new types of clinical trials. And together with Dan Hayes, we called them post-adjuvant clinical trials to show that at the time point when we see a molecular relapse indicating MRD, can we design interventional trials to show whether we can cure the cancer patients even at that stage before they develop full-blown metastasis. And that is a big question in the field right now. Well, how can we detect? What do we know about the detection of MRD and post-surgical surveillance? And I will focus on the ctDNA today. Well, there is actually excellent paper in different types of solid tumor where people have used very sensitive technologies to detect such molecular relapse. For example, here in lung cancer, the group of Max Steen, which has shown if patients had patients that had never detected any ctDNA had an excellent prognosis here compared to the patients where ctDNA was detected at the beginning or during follow-up. And I think this is a small analysis of only 40 patients, but very promising data. Another study was done by the same group in the context of immunotherapy, which has, of course, changed the therapy landscape in oncology over the past years. And also here, the group could actually show that the detection of DNA was a good prognosis, was a bad prognostic indicator within this group of patients receiving immunotherapy, and it may indicate early on which patients will and which patients will not profit from the therapy. And since the therapy has also side effects, that's important information, but again, a small study that needs to be confirmed. In breast cancer, the group from Charles Coombs and Jacqueline Shaw has actually published this very nice study, also following up patients with breast cancer. 49 patients were investigated, And again, the patients that had no ctDNA detected had a very good prognosis compared to the patients where ctDNA had been detected. And interestingly, this molecular relapse was detected hundreds of days before imaging could actually see a relapse. And this lead time may give us now time to really start with therapy earlier. And these are the studies that need to be done in the future in clinical trials. We also have data using circulating tumor cells. Here, for example, we performed a study 2 years after initial therapy, and we could show that the patients that had no circulating tumor cells, the blue lines, had a favorable prognosis compared to patients that had CTCs 2 years after initial therapy, the red line. And of course, now we have different liquid biopsy markers that can indicate that there is residual disease and may allow us to detect relapse much earlier. One of the key applications of ctDNA analysis is, of course, to get information on therapeutic targets and resistant mechanisms, and this, of course, has a clear impact for the management of cancer patients. And of course, if we look at tumor DNA, we ask, can we see genomic aberrations that are relevant to the choice of a therapy? And in addition to that, we can also look at tumor cells where we can analyze transcriptional changes in relation to therapy response. As you can see here in this very recent review that I wrote with Katerina Lixpan-Abier from Montpellier in the 10th anniversary issue of Cancer Discovery, there are many, many drugs that can be given based on certain mutations that we can now detect now on the circulating DNA, as shown here on the left side of the graph, in different types of tumor. So this information has some relevance for the management of cancer patients. And in addition to that, we can analyze cells for transcriptional changes that are also relevant to therapy. We have done that, for example, with the GenoMass array system here in a population of very difficult patients. These patients had non-small cell lung cancer, known to have low ctDNA concentrations. And on top of that, these patients had brain metastases, and brain metastases are known to shed not so much DNA, maybe due to the blood-brain barrier. But we were able here with this panel that looks at 5 genes and 67 hotspot mutations to detect actually ctDNA in these cancer patients. And we could also follow up these patients and could see whether there were emerging new mutations that were arising in these cancer patients. Overall, we were able to get informative ctDNA from around half of the patients analyzed, and I think most of those patients had also oligometastatic brain metastasis, so brain metastasis only, and also these patients, the detection rate was around 50%, which I think is very nice. And the CTCs that we analyzed in addition gave a little bit of additional information, but most information was derived from the ctDNA analysis. We also analyzed actually whether we could find more CTCs in these patients and then do it in conjunction with the ctDNA analysis. And this is work from Harriet Wickman's group in our institute, showing that if you select these CTCs, not just based on EpCAM, but on HER2, HER3, and EGFR, that we can increase the capture rate. So maybe in the future, we can combine the ctDNA and CTC analysis in these patients more efficiently. Of course, what is also very relevant is that we actually monitor cancer evolution that is actually driven by the applied therapy. And this example from Alberto Bardelli's group, they looked at patients with colorectal cancer, and they received targeted EGFR-directed therapies. And they could actually see here in this curve that initially during the therapy, the ctDNA level dropped to 0, and it remained 0 for several weeks. But then at week 14, several different clones actually emerged, not only the original clones, but additional clones emerged here. And this is a very good example of how we can measure acquired resistance to therapy by sequential DNA blood analysis. Another example by the group of Jackie Shaw in breast cancer, they actually looked at mutations during endocrine therapy in breast cancer. They looked at the blood at the baseline level 4, 7, and 12 months after initial therapy, and they could actually show that initially they did not find any resistant mutations, but over time these resistant mutations emerged, and that is a very nice example how monitoring circulating cell-free DNA can give you information on resistance to therapy. The future, in my view, is to combine the ctDNA analysis with the CTC analysis. And of course, immune checkpoint inhibition therapy monitoring is one of the key applications. Here we can look at protein expression on the CTCs, like the MHC expression relevant for the recognition of the tumor cell, the PDL expression relevant for the inhibition of the T-cell response, and we can look, for example, at tumor mutational burden at DNA and combine this information. In Hamburg, we have actually established a very active group that is focusing on melanoma, the Northern Germany Liquid Mel Biobank Consortium, and we have in particularly very good young researchers that were supported already by awards that are very active in this group. We have actually shown that ctDNA is actually increasing in increasing stages of melanoma, as shown here on the right side, and the same way CTCs, but ctDNA was a bit more sensitive in that regard. We have then also, analyzed and captured the circulating tumor cells, picked them, and analyzed them for therapy-relevant mutations, such as a BRAF V600 mutation. And we are now actually have a new research project together with Agena that we try and get this genomic information without the need of whole genome amplification, which can always create a bias in this single cell tumor analysis. And we have very promising first results. And this is a workflow for the mutation analysis with the Agena system without the need of whole genome amplification. We actually pick the single circulating tumor cells, then we do a targeted amplification of the locus of interest, and then we analyze that DNA with the mass spec system. We actually do that in the context of defined clinical studies. In metastatic melanoma patients, we have here a sequence of blood analysis every few weeks, and we analyze the circulating plasma EDTA for circulating tumor DNA, but also the CTCs and additional liquid biopsy markers. And at the end, we want to get a multi-composite biomarker panel that includes different liquid biopsy biomarkers. And we also focus really on the single cell and the CTC and ctDNA analysis. In patients with adjuvant treatment, which are patients where the tumor is resected, we also have a very dense sampling of plasma for the ctDNA analysis. And here we ask the question, can we identify patients early enough that profit from this treatment? And can we also identify patients that do not profit from this treatment and can be spared the side effect? And I think particularly in the adjuvant patients, this is a very important question. Well, so in conclusion, the liquid biopsy provides information on the dynamic tumor evolution in individual patients. And I think that is complementary to the information obtained by tissue biopsies. The assays need to be standardized and validated, and we have established such consortia to do that by independent expert groups. And I think the future is that we need interventional clinical studies to demonstrate clinical utility of liquid biopsy tests. That is very important for regulatory approval. And in these interventional clinical studies, we have to show that an intervention, a therapeutic intervention, for example, based on our liquid biopsy blood analysis, has actually benefit to the cancer patients by, for example, switching the therapy from a less to a more effective one. I also should say that I'm part of the ESMO recommendation group that currently writes the guidelines for the ctDNA analysis, and I think writing these guidelines is also a very important help for this field. So if I sum up now from what I've said, I think we have, of course, applications for the ctDNA analysis as screening and early detection of cancer. We can use it for staging and monitoring of localized cancers. We can get information on the prognosis and risk assessment and see which patients have a higher risk to progress and do a real-time monitoring of therapies. And of course, we can track tumor evolution under therapy, and I showed you a few examples of that. And the future is, of course, to get all this information together now in the design of interventional clinical trials. And these interventional clinical trials, and here are a few examples given, they will actually provide the clinical utility required to get liquid biopsy assays into clinical practice. At the end, I would like to thank our Liquid Biopsy Research Network at the University Cancer Center here in Hamburg, and it consists of research institutes on the left sides and clinical department on the right sides, treating patients with diverse types of solid tumors. And this is a very, very successful interaction between basic and clinical research. I would also like to thank the people in my institute. We have 40 to 50 people from 13 different countries, a little bit like United Nations. And I would like to thank the agencies that support our research, and particularly the European agency. I got 2 advanced ERC grants and also funding from the EU IMI, and also the German agency like DFG, BMBF, and Deutsche Krebshilfe, who is supporting our research for more than 20 years now. So with that, I would like to finish my presentation and hand over the presentation to Dr. Alexander Sartori from Agena Bioscience. It's all yours now, Alex.
Yeah, thank you very much, Klaus. Thanks for your kind introduction. And thank you, Klaus, for your wonderful overview and comprehensive overview about this exciting field of liquid biopsy, and especially focus on ctDNA and its clinical relevance. I mean, in your talk, in Klaus' talk, we've seen a lot of data, and we've heard about some projects and published data that were generated with the MassRay. And for those who don't know this technology, I will take the opportunity to make you a little more familiar with what Agena can offer and how the MassRay works. Again, my name is Alexander Sartori. I'm at Scientific Affairs at Agena. And for those who don't know Agena Bioscience, whose mission is to deliver affordable, targeted genomic testing to the clinical labs around the world. So Agena is headquartered in San Diego, California, but it operates sales offices in other regions of the world, such as in Brisbane, Australia, Shanghai, China, and Hamburg in Germany. And our advanced genotyping platform delivers timely, accurate, targeted results to improve decision-making and laboratory economics. So our technology is the MassRay system, which is a mass spectrometer, which is for analyzing nucleic acids, so DNAs and RNAs. I want to show you the simplicity of the workflow that the MassRay offers. So it actually starts from a very simple wet lab workflow that is PCR-centric. This is consisting of a real PCR amplification and a single base extend reaction, which you can also compare in terms of simplicity to a PCR. This entire workflow takes a couple of hours, but the hands-on time of this wet lab step is only marginal. It's only in the range of 20 to 25 minutes. After the wet lab, after your reaction products have been analyzed or have been created, now it's time to analyze those on the target sequence detection unit, which is the MassRay system. This is a fully automated system and a fully automated workflow that runs about, depending on the application and on the throughput that you're having, in the range of an hour to 90 minutes. But again, also here, because it's fully automated, it's only 5 minutes hands-on times are required. And when it then comes to data analysis, this is really the simplest. So as Klaus mentioned in his talk, no bioinformatics is required. And it's really easy data interpretation. We provide a number of reports actually for each application that you are running there's a report available that provides you data on a single click. So to summarize, the total elapsed time after for the entire mass array workflow, including the wet lab steps, is between 8 and 9 hours. Again, this depends on the throughput of your application and on the actual application. And the totally required hands-on time is below an hour for that. And again, no library prep is required to generate your data, no size selection prior, and no bioinformatics pipeline is required. The middle component, that was the target sequence detection. This is the mass array system. And I don't want to go into deep detail about this technology because it's a fully automated system and it does the work for you. The full automation starts, what is shown here as the number 1, from the liquid handling of your samples coming out of the wet lab step. And then after liquid handling, the what we call a chip is transferred automatically onto the mass spectrometer itself, where the data acquisition is also happening, fully automated. This technology allows you to run multiplexed analysis of hundreds to tens to hundreds of genetic targets at the same time and in the same sample. And as shown before, the turnaround time can be achieved in a working day or overnight. This includes completely easy data analysis and provides fast results to make critical decisions. And in addition to its simplicity and easy workflow, this method is cost effective, and the testing costs you a fraction of the cost of NGS or other technologies. So with this underlying technology that is universal for all the applications that you see here on this slide, namely pharmacogenomics, SARS-CoV-2 testing, sample integrity, and more, today we want to focus on, we are focusing on oncology, so we're certainly also offering applications for tumor profiling, and in this particular case, for liquid biopsy. When it comes to analyzing oncology samples, so we are looking for somatic mutations, we require, and Klaus also mentioned this, we require a super superior sensitivity. So for interrogating somatic mutations in tissue, the chemistry that we call IPLEX-HS chemistry, and this is just a trade name of the biochemistry that we are using for tissue material, reaches a limit of detection down to 1%. And when it comes to applications that require even higher sensitivity, such in liquid biopsies, we are using the ultra-seq biochemistry with another magnitude of sensitivity. So we can go down to 0.1% minor allelic frequency with this chemistry. And what these two applications have in common is that you start from a single multiplex PCR reaction. So you only enter your precious sample once into the reaction, and it generates all the amplicons that you require to interrogate the tens or the 100 mutations that you're interested in and that are clinically relevant at the same time. And they both have in common that you need very little input material. So we recommend to enter as little as 10 nanograms of DNA for your analysis to reach the required sensitivity. As topic of today is liquid biopsy, I want to focus and show you a little bit of the offerings that Agena has in this field, in the field of liquid biopsy. And again, our brand name for this biochemistry is UltraSeq. And in this area, we offer non-small cell lung cancer panels. One is a focused EGFR panel that contains the most clinically relevant 6 EGFR markers, the main activating mutations in EGFR and the major resistance markers. But if you want to have a bigger picture, you would use the lung panel that covers more than 70 clinically relevant variants in 5 oncogenes, namely BRAF, EGFR, ERBB2, KRAS, and PIK3CA. Leaving non-small cell lung cancer behind and going to the colon, where our panel covers more than 100 clinically relevant variants in 5 genes: BRAF, EGFR, KRAS, NRAS, and PIK3CA. And in the field of melanoma, and also Klaus mentioned 2 studies where we were using that panel, we interrogate more than 60 clinically relevant variants across 13 genes. And again, all these variants in these panels can be distinguished from each other. So you can not only say it's an EGFR exon 19 deletion, but you can say which one it is. This is the portfolio that we are offering from the shelf. And because you may want to start your analysis from a tissue biopsy, we certainly also offer content overlapping panels for monitoring mutations or for detecting mutations in tissue. These are not shown here, but these are very, very similar to the ones that are shown here. And if your research requires other applications, we do have a custom design service offering that is called Assays by Agena. And this service can create any panel for any application that you can ever think of. Any targets can be analyzed and realized by our assay design service called Assays by Agena. When it comes to, when we want to go back to the technology, the mass array system itself using the ultraseq biochemistry for a liquid biopsy, Klaus was already mentioning this publication from Cancers from last year by Weber et al., where in the Cancer ID Consortium, we were comparing the mass array system to other technologies. And he already mentioned the superior precision of the technology. So in the picture in the middle, you can see that the coefficient of variation was the lowest with the mass array when comparing to other technologies with comparable DNA input amount. And the plot to the right is showing how close a known sample, or a sample with a known allelic frequency, in this case of 1%, is detected across replicates with the various technologies. And you can see here also that the mass array has the lowest variation across the technologies and is only beaten by digital droplet PCR when adding much, much more DNA. So in this case, 70 nanograms rather than 12 or 15 nanograms that was used on the mass array. In another study, the authors actually were comparing the ultraseq lung panel to the FDA-approved cobas EGFR version 2 test. That is the gold standard for EGFR testing. This study was also published in Cancers in October last year. And on the graph A, what you can see here is that there's a high concordance between the cobas EGFR and the ultraseq lung panel that is depicted by the blue portion of the pie chart. So correlation was almost 80%. But you could, and this is thanks to the superior sensitivity of the ultraseq technology. The green piece of the pie, representing a bit more than 14%, is the extra content, or are the mutations that mass array finds in addition to cobas, thanks to the superior sensitivity. And in plot B, we were expanding this comparison towards all clinically relevant mutations. So the cobas EGFR certainly only covers EGFR content, but thanks to the broader menu of the ultraseq lung panel, that also has BRAF, HER2, KRAS, and PIK3CA content, we could detect even 25% more clinically relevant markers or mutations. So, I want to come to an end, and not without telling you that we are continuously extending our portfolio, and I want to give you a sneak peek preview to what we are doing currently. So I was showing you the existing liquid biopsy panels, so non-small cell lung cancer, colon, melanoma, but we're extending that towards an alpha-seq breast panel. And this is currently under developing, and we expect it to be available next year. However, we certainly always work with collaboration partners such as Klaus Pantel in the UK in Hamburg. And for a study that was just published very recently, in August this year, we were using an early custom version of the AltraSeq breast panel to comparing breast cancer to brain metastasis. And if you're interested in these results, just this study is now publicly available. And because in particular PIK3CA testing becomes more and more relevant in breast cancer, as you see on the left-hand side, 40% of patients with hormone receptor positive and HER2 negative advanced breast cancer have a PIK3CA mutation. GINA is now preparing for launching a specific PIK3CA panel for tissue testing, and that is coming very soon to the market. That covers 20 clinically relevant markers in PIK3CA and has a sensitivity of the highest of the markers with the highest frequency of in the range of 1% allelic frequency. We could show in a recent ring trial that we participated in Germany, and that was not tissue, but it was done on liquid biopsy, but we were using that prototype of that panel, of that application, that we were outperforming many other technologies. So in this ring trial, only 13 labs were participating, but only 4 were passing the ring trial successfully, and 2 of them were using the MassRay system and the panel that I was just mentioning. So yeah, with this outlook about what's coming, what is available, and how the technology works, I want to come to an end, and I think we have ample time for your questions. With this, I want to thank for your attention already.
Thank you, Dr. Sartori and Dr. Pantel. As a reminder to attendees, you may type in a question at any time using the Q&A panel, which appears on the right side of the presentation. We will now start the Q&A portion of the webinar. The first question is for Dr. Pantel. Which technology was used to combine cell search and HER EGFR at the same time? HER EGFR analysis, I presume.
Well, actually, it was not done with the cell search. It was done with Miltenyi beads, where we combined these antibodies to these HER3 and EGFR in a test together. It was not the cell search system.
Thank you. The next question, also for Dr. Pantel. Is any type of circulating tumor DNA reference material used during the validation stages of the developed tests?
Yeah, I think this is a very good question. It was in Cancer ID, we also had a custom supplier of reference material. I think it was Seracare. And we actually added these defined, pieces of DNA in our ring experiments. And this is a very helpful tool, actually, to develop assays and also test the proficiency of different labs by using these defined DNA pieces with defined mutations. And I think I can highly recommend that for ring trials and proficiency testing. Yeah. Mm-hmm.
Thank you. The next question is also for Dr. Pantel. Is the size-based isolation performed using Parsortix technology?
Well, we have actually used that technology also for many years in our labs, and it's interesting. We see some cancer patients that have higher CTC counts with the Parsortix, and other patients that have higher count with label-dependent systems like the cell search. So it seems to be that these size-dependent technologies can be very complementary to antibody-dependent technologies. But it's not that one is better than the others. I mean, the hallmark of tumor cells are the heterogeneity, and the heterogeneity is true for the expression of markers, and the heterogeneity is true for the size and deformability of these tumor cells. So in our hands, really, both technologies have advantages and disadvantages. And the combination of technologies, let's say if you have a CTC negative sample with one technology, try the other technology, whether you are able to capture cells probably makes sense.
Thank you. The next question is for Dr. Sartori. How much plasma volume is required to extract 10 nanograms of circulating tumor DNA?
Yeah, that's very relevant. In the clinical practice, you certainly want to make sure that that you can analyze samples that fit to the routine. So when 10 nanograms is what you usually get from a single blood draw. So a single blood draw means 10 milliliters of EDTA blood or coming from a Streck tube that contains between 3 and 4 milliliters of plasma. And when you extract the cfDNA from that, you will, in most cases, you will get more than 10 nanograms, so you even don't have to use up the entire sample.
Thank you. The next question is also for Dr. Sartori. Is the MassArray DX a laboratory-developed test or FDA regulated?
Well, the MassArray DX itself is the technology that has the diagnostic. So it is an approved technology, is an approved system. It can be used universally for all applications that run on it. So it's acquiring data for all applications, and it can run any lab-developed test that is designed for the MassArray or for any validated test.
Thank you. The next question is also for Dr. Sartori. Does Agena have any exosome, I believe this would be exosome analysis or isolation devices for research in its portfolio?
We currently don't. So, I mean, the question was if we have any exosome isolation solution.
The question is, does Agena have any exosome strategy device on research on it? But I believe,
okay. So we currently don't. However, our focus is ctDNA analysis. But as in the presentation of Klaus, we could see we are always looking into expanding our portfolio. So he's shown some very exciting and early on data on ctDNA. And he mentioned, exosomes, for instance. We are currently not looking into it, but we're always open to expanding our capabilities.
Thank you. The next question is for Dr. Pantel. What is the average concentration of circulating tumor DNA in early stage and advanced stage patients?
Well, that depends largely on the tumor type and also on the site of advanced metastasis. For example, if patients have only brain metastasis, oligobrain metastasis, the ctDNA concentration is lower than if the patients have, for example, liver or bone metastasis. But let's say as a rule of thumb, if you have an early stage cancer patient, your technology should have a sensitivity clearly below 1%, because it's certainly that most patients have below 1% ctDNA concentration. If you have metastatic patients, it can be above this 1%. But again, there's large variation from tumor type to tumor type and also from the site of metastasis. But just as a rule of thumb, if you have a technology that has a detection, let's say, lowest level of 1%, and you go for early stage cancer patients, most of your patients will have not sufficient amount of DNA. So very important to choose the right technology for the right stage of your disease. And if you are unsure, please make a small pilot trial to see whether you measure anything before you plan a large clinical trial.
Thank you. The next question is for Dr. Sartori. For the mass array, what input material is required for starting the workflow? It is suggested here 9 milliliters of cell-free plasma with a question mark.
Well, I mean, input material would be the extracted DNA, whether it's genomic DNA or this question is referring to plasma, so would be extracted circular cell-free DNA, so cfDNA from the plasma. This question suggests 9 milliliter of plasma. This will in most cases contain more than required, more than necessary amount of cfDNA. Again, we recommend to start with 10 nanograms. And in this context, I would like to mention that it's really, in our opinion, really important to do proper upfront quality check of the samples, so an upfront QC to really assess the copy number of the sample, so how much DNA is in your sample, and is it degraded, or does it have any contamination of genomic DNA that is not wanted, and that would lower your sensitivity. So before running a liquid biopsy test, our clear recommendation is to use an upfront QC method. And there's many available in the market, and there's also certainly an option on the mass array to properly QC your sample upfront.
Thank you. The next question is for both panelists. Can liquid biopsy replace tissue biopsy, or should they be tested simultaneously?
Well, let me start. I can start maybe. No, I don't believe it will replace tissue biopsy totally. I think tissue biopsy, of course, can also give you some information on the composition of tumor cells, subtypes. It can also give you information in addition to the DNA mutations. And sometimes you may get more material by the tissue biopsy than you can probably get out of the blood. But I think that, for example, for sequential monitoring of therapy responses, over time, there is no alternative to the blood, actually, because nobody wants to have repeated needle biopsies within a short time frame, And I think here the blood is certainly very, very helpful to get really real-time information and get sequential information on what's going on during therapy. But let's say for the first analysis, when a patient comes to the doctor or to the clinic, I think, of course, everybody will look at the tissue. So I don't think that the tissue will be totally replaced by liquid biopsy in the future, but these technologies are clearly complementary.
Yeah, and if I may add something, there are certainly cases where you can't obtain a tissue biopsy for various reasons because of the health condition of the patient. And there are studies available where it was shown that results from tissue and liquid biopsy were comparable. And then if you don't have a tissue biopsy available, then it would be always a good idea to go ahead and test the liquid biopsy.
Thank you. The next question is for Dr. Pontel. What sensitivity do we need to detect minimal residual disease using liquid biopsy?
That's a very good question. For example, it depends also on the tumor type. For example, in breast cancer, we have seen that you need to go down to 0.01%, that it can be very low. So, here I think we probably need very, very sensitive technologies. However, you can also say if your technology is not that sensitive, let's say you only have 0.1% lowest detection, you will also still detect a molecular relapse earlier than the imaging, So there are arguments that some people say, okay, you don't need a super, super sensitive technology because then you may run into specificity problems. But if you have a sensitive technology like 0.1% lowest level of detection, you still detect your MRD months before you do it with the imaging. So the question can be answered that way. You definitely need a technology that is going below 1%, whether it needs to go below 0.1% or whether it needs to be much, much more sensitive than 0.1%, that can be debated.
Thank you. The next question is for Dr. Sartori. What is the throughput of the mass array? How many liquid biopsy samples can be tested?
So the throughput of the mass array is basically not the bottleneck. The mass array can process a lot of samples. It is you can call it a working horse. The throughput or the limitation would rather be how many PCR cyclers you have in your lab. But if you talk about a single PCR cycler and you're getting 20 liquid biopsy samples in a day, this can be achieved. On a single run, though, with interrogating in the range of 70 to 100 markers from a single sample, on a single run, we can process up to 8 patients at the same time. However, the instrument can handle 2 chips, and so we can double the number, but multiple of these runs can be performed per day, if this gives you a perspective.
Thank you. Our next question is for Dr. Pantel. Are there significant differences in ctDNA concentrations between different tumor entities?
Yes, there are significant differences. I think this has been already published many years ago. You have tumors such as primary brain tumors, which have low ctDNA concentrations, probably due to the blood-brain barriers. You have other tumors like colorectal cancer, where you have very nice ctDNA concentrations. And then you have non-small cell lung cancer, which is a little bit in between the good and the bad ones, so to say. But I should also say there are considerable intra-tumor heterogeneities. So it's not that all patients with the same type of tumor have the same DNA concentrations, even if they are at the same tumor stage. So there's marked variation, and we still do not understand very well which individual tumor is a good shedder and which tumor is a poor shedder of DNA. We need really more research to understand why some of those patients have higher and other patients have lower DNA concentrations. But overall, yes, there are differences between tumor types and locations of metastasis.
Thank you very much. That is all the time we have for today. If we didn't have time to get to your question, we will try to follow up with our experts after the webinar. We'd like to thank Klaus Pantel of University Medical Center Hamburg-Eppendorf and Alexander Sartori of Agena Bioscience, as well as our sponsor, Agena Bioscience. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you very much for joining us for this Genome webinar.